Okay, well, let's move on to our last talk today. I think we're on time. How is this ever possible? We're never on time, but we're perfectly on time. And Clint is going to give our next talk. There's his title. That's great. That helps me. For those that don't know, Clint is the PhD student in Ava Top's lab. Clint, I'm sorry, I can't remember if this is your third year. I think that's right. Fourth, dang, I did that last time too, didn't I? Fourth year, so that means he's about ready to defend and get out. No, maybe, all right. I like that too. No, Clint's been a great student in the BCB program, and we're really excited to get another update from him on some of his work. This is not rotation work, unlike the last couple of our talks. I don't think it is anyway. And so Clint's going to talk to us today about using gene co-occurrence to discover a novel anti-phage system in Vibrio cholerae. Take it away, Clint.
So two things. This was a Beacon collaboration, but I did do it in my BCB rotation. So yes, I wrote most of the software in a rotation with Dr. Ben Ridenour as well.
That's
so I also think we need to put Kristen at the end of these IBEST lunches, because I don't know how I'm going to follow up on hairy selection and space alien combat. But yeah, so I'm here today to share with you how I use gene co-occurrence to help discover a novel anti-phage system in Vibrio cholerae. There we go. So Vibrio cholerae is a waterborne bacterial disease. It infects 2 to 4 million people annually, and there is no definitive cure. You often find it in areas without potable water, or in areas after natural disasters or warfare destroy infrastructure. And we're currently in the 7th pandemic of cholera, and each pandemic is marked by a new strain that has outcompeted the old strains. And so in the current pandemic, it's the L-Tor strain that emerged in 1961 that is the dominant strain. So as biologists, we're very interested in why is this L-Tor strain so successful? And as many of you may know, bacteria can carry horizontally acquired regions of DNA called genomic islands. And these genomic islands are often full of virulence factors. And in fact, L-Tor has 2 novel genomic islands. They're named Vibrio 7th islands 1 and 2. And this distinguishes L-Tor from other strains of Vibrio cholerae. So the thought is if we can figure out what these 2 genomic islands are doing, maybe we can figure out why L-Tor has become the dominant strain driving the 7th pandemic of Vibrio cholerae. And so there are 2 genomic islands. For this presentation, I'm going to focus on VSP1, which has 11 genes. So Dr. Chris Waters and Dr. Jeff Severance are researchers at Michigan State University, and they discovered the function of 2 of the genes in the first VSP1 genomic island of L-Tor. They're CAPV and DNCV, or they encode those. And the first protein they encode, the DNCV, makes a secondary messenger, which activates the second gene product, CAPV, which is a phospholipase. And it actually eats the cell wall of Vibrio cholerae and makes the bacteria smaller. So you get a smaller plate morphology, which you see here on the left with L-Tor, as compared to the classical Vibrio cholerae on the right. So this is very exciting because this is the first time that the genomic islands were tied to an actual phenotype in L-Tor that might help explain its dominance. And buoyed by this success, they were looking for a bioinformatic analysis of these 2 genomic islands to help them get some direction on which of these genes may work together. There's a total of 30 some odd genes in both islands. And so they turned to Beacon. Many of you may be familiar with Beacon. It's an NSF collaboration to study evolution in action. And I got on board to help them with the bioinformatics. So we've got these 2 islands, and they're unique. If you blast them, you get no hits. And so my thought was that we could try to predict gene networks from each island. And for my purposes, I consider a gene network to be a set of genes that are functionally related that work together to accomplish a task. So for example, the CAPV and the DNCV, the genes that encode those are a gene network that help give you small colony morphology. And what I'm showing on this page is an overview of what the software I wrote, Correlogy, does. It takes a novel genomic island, like the 11 genes in the top of the VSP1, And what you get as output is a visualization of a putative gene network. And each gene becomes an oval, and the strongest relationship between genes is shown. So this is actual output from my software. And one of the things you'll notice is it actually accurately reflects the relationship between CAPV and DNCV that the Waters team had previously discovered. So how do we do this? How do we go from a set of genes just in a row in a genomic island to a visualization of putative gene networks? Well, the idea is that we use co-occurrence across the bacterial domain. So the idea is that genes that work together move together. So for example, again, looking back at VSP1, these two, the genes that encode these two proteins, CAPV and DNCV, homologs of those are going to be found at a much higher frequency co-occurring together than they would be with genes that are not in that gene network. And so Kim and Price are a team who looked at this quite a bit in 2011, and they coined the term Correlogs, which is a mashing together of the words co-occurrence and homologs. And what Correlogs really is considering is the favored or non-favored co-occurrence of a homolog of genes across the bacterial domain. So how do we get this Correlogy score? I'm going to use the VSP1 island from LTOR as an example. You would take all 11 genes, and you can do this in my Correlogy software, and the first thing you would do is you would blast each gene for homologs. And then you would merge, and then you merge those blast results into a presence-absence matrix. So, and the columns are the genes, and the rows are any bacteria in which at least one homolog was found. So for example, if you look at the bottom of this matrix, Acetobacter, a homolog of gene 175 was found in this Acetobacter species, and a homolog of gene 176 was found in this Acetobacter, but not homologs of these other genes, no other co-occurrence. So from this presence-absence matrix, we calculate a Pearson correlation from each gene I to J. And then over here, I do another partial correction, which is something Kim and Price came up with to help take care of the transitive effect. But what you end up with is a WIJ score, and we call this a Correlog score. And it is normalized between -1 and 1. And what a Correlog score of 1 means is that the homologs of two genes always co-occur in bacterial species that we're considering. And a score of -1 means homologs of these two genes never occur in bacterial species that we're considering. Whereas a score of 0 means they co-occur at a frequency that you would expect just randomly. And since this is an I to J calculation, what you get, for example, for gene 175 is a value from gene 175 to all the other genes in the genomic island. And you get a maximum score. For example, 175, the highest Correlog score is to 179. And we can use this maximum score to create a visualization of putative gene networks, which is what I'm showing you here. So in this visualization, each gene is a node. So for example, on the left, 175 is a gene and it is a node. And when we look back at the matrix chart, we see that 175's highest Correlog score is to 179. So that highest Correlog score becomes an edge, which you've seen here, pointing to 179 with the Correlog score there. And so what was Correlogy able to predict for VSP1, the genomic island, in 7th pandemic LTOR? Well, it reflected two things that were already known. The first thing was the relationship between CAPV and DNCV. Again, this edge is the maximum Correlog score, and it predicted that relationship that the Waters lab had discovered that causes the small colony morphology. Another team had built on the Waters findings and found out that those two genes are actually part of a larger system called CBAS. And you'll notice that Correlogy also correctly predicts the relationship of the genes within CBAS. And CBAS is this interesting system where if an LTOR bacterium is infected by a phage, it actually activates the CBAS system. This CAPV phospholipase eats the cell wall of the LTOR bacterium and kills it. It's called altruistic autolysis. And so the phage are not able to successfully invade LTOR populations because as soon as an LTOR bacterium is infected, it kills itself and the phage can't successfully invade the population. So those two things were known, and it's exciting that they were correctly reflected by Correlogy. But the third discovery that Correlogy made is the relationship between gene 175 and 179. And remember that the product of 179 creates a secondary messenger chemical. So when I relayed this to the Waters team, the thought was that 175 might be a protein that's activated by the secondary messenger molecule. So the Waters team ran down this Correlogy prediction of the relationship between 175 and 179. They spent 2 years discovering that the function of this 175 gene, that it encodes a protein, and this protein is a deaminase, and they named it DCDV. And in fact, these two genes, 175 and 179, are a novel cytidine deaminase-based anti-phage system. And not only is it found in Vibrio cholera, it's also found in many bacteria across the bacterial domain. So this was pretty exciting. And in fact, they found out through their wet lab analysis that this DCDV protein is in fact activated by the secondary messenger molecule that's produced by the protein encoded by 179. The other thing I would point out about the strength of Correlogy in this case is that a lot of times when you're looking at the relationship of genes, you might look for genes that are under the same promoter in an operon or are right next to each other. But in fact, if you look at the top of the genomic map of this, these two genes that make this novel cytidine deaminase system are separated by 3 other genes, and they're expressed in the opposite direction. So this relationship between these two genes is not, exactly obvious, whereas Correlogy was able to link them together and give the lead for the wet lab team to discover this stuff. So my takeaway is that Correlogy is a software package that predicts functional gene networks in novel genomic islands. It was used to help lead discovery of a novel anti-phage system in 7th pandemic L-Tor Vibrio cholera. And in fact, it probably is the lowered susceptibility to phage infection that makes L-Tor successful. And this novel anti-phage system is found in many bacteria, in addition to Vibrio cholera. So with that, I want to give my acknowledgements. Like I said, Dr. Ridenour helped me on my rotation, helped me with the math. I was just learning the program, very helpful. Dr. Top, my PI, who made me do the Beacon collaboration, even though I didn't want to, and I'm very glad I did now. It was an excellent BCB project. And Dr. Brown, who helped me at the very beginning before she retired. And of course, the people at Michigan State University, the Waters team, who spent 2 years. I just handed them the predictions and the analysis. They did all the hard work of cloning the genes. So the first 2 lead authors here are co-authors, Jeff and Brian. Huge amount of credit to them for their hard work. And this paper has been submitted to Cell for publication. So with that, I will take any questions.
Super cool. Mohammed has a question in the chat there. Clint, how did you assess the prediction accuracy?
So that is something that I'm actually working on with Dr. Ridenour. So I got it right with Vibrio cholera, but that's an N of 1, right? So what we worked on, what I worked on my rotation and what I'm going back to now is we're taking known gene networks in the KEGG database, and we're pretending like we don't know the relationships, and then we're testing Correlogy against that. And one of the things we want to do is right now we're using a Pearson correlation, but there are other similarity methods and distance-based methods, and we want to try these different ones to see if there is something better or more accurate than the Pearson correlation with the partial correction.
Cool. I have a quick question, Clint, about that first step in your, was a BLAST step, and obviously BLAST has parameters associated with it, and having done lots of BLAST, you get all sorts of hits, sometimes that are are not necessarily homolog. So how do you limit that so that you're actually getting the right stuff, and maybe more importantly, not missing other things?
So that's a really good question, and the answer is that it's tricky. Like in all bioinformatic analysis, a lot of times you have to pick parameters, yeah, and the parameters you pick can be subjective, right? Yeah. So I did BLAST P, and I did an e-value of 10 to the negative 4, which usually that's kind of a back of the envelope number for functional relatedness. because ultimately we're looking for genes that are functionally related. We actually don't want genes that were vertically transmitted, right? So I excluded the Vibrio cholera from these that have these islands, because the idea is to track the gene networks. So, and you also don't know if they're paralogs or orthologs, but the power is from the big data, right? The hope is that by using all 250,000 bacterial species in NCBI, as long as you get enough blast hits, the power of that will overcome the kind of things you're talking about. And I think my presence absence matrix had like 28,000 species hits. So it seems like a pretty high number, but it's one of the things that also needs to be tested along with the KEGG test set and different distance matrices.
That makes sense. I wonder, because reducing that down to presence absence, that's you lose information. You're taking some amount of matching between these, between things in the database, and you're saying, well, it's all the way down to presence or absence. I feel like there's the opportunity to miss stuff. And I was trying to think, like, is there a gene tree way to do this, or is there any benefit to doing this with a gene tree?
So there's another team that used the Kim and Price approach that I also used, and you actually input the phylogeny with all of the genomes. Mm-hmm. And the idea is that within the set of genomes and phylogenies that you included, it looks at relatedness.
Oh, cool.
One of the things to remember here is we're looking at horizontal transfer.
Yeah.
Right. So it's kind of trickier. You don't necessarily want to include phylogeny. You want the weight of the whole bacterial domain, because these genes could be present anywhere and they could be rearranged. And like I said, if a set of those genes ended up in different places, but they're still functionally doing the same job, they'll stay together. Right. So.
Yeah. Cool. Other burning questions?
Clint, did you mention that you didn't want to do this rotation originally? I mean, to me, it sounds really cool.
Well, so I shouldn't have said that. Not the rotation, but the Beacon collaboration. And the truth is, I really wanted to do it because I was like a first or second year student. And then by a year later, I was like, why am I doing this? I'm not going to get any papers out of it, And then I heard back from this team 2 years later, and they're like, hey, we ran with that and we found out this super cool stuff. So now I'm actually rewriting the software that's on GitHub, because it's total trash. I wrote it when I just learned the program, and I'm going to do all these test sets and try to publish it. But you never know. It's hard to predict what is fertile land for research sometimes.
Yeah, that's super cool.